Thank you so much for everyone is coming to listen my the research project, what I did for my previous PhD project and my postdocs. So basically, I'm community ecologist. Basically, my expertise is on biodiversity and climate change. So basically, I'm talking about the two aspects. One is the community structure in metacommunity, because the habitat is being fragmented, so that we are going to look this biodiversity in the broader context that is called the metacommunity. So I will give a little bit very brief background about the metacommunity and the research finding what I did. And the second portions I will talk about the climate change and the biodiversity, basically mammals. About how we can improve the biodiversity model in terms of the climate change, and then what will be the mammals biodiversity in future in a global scale. So basically, I'm looking at the global scale, the regional and the global scale. Let's talk this one. We know that this is the picture showing that the habitat is being fragmented. So not only the terrestrial ecosystem, also in the aquatic ecosystem, habitat is being fragmented either by the dam or pollution or eutrophications. The different processes, the habitat is being fragmented. So as a community ecologist, we are interested to see what processes save the community structure. How many? That means what the species are there, how many species are there, or what are the relative abundance of the species in the communities. In general, in the community ecology, we focus on a single scale, assuming that all the community are close and isolated. Just like what we did we have an idea about the Lorca-Volterra model, assuming that there is no influence from the outside. But in reality, there is a connections between the communities. In the situations, the traditional community does not explain the influence of the dispersal or the migration in order to find the structure, in order to find what the process structure our community. We know that the community structure is from the island biogeographic theory. The number of species are based on the size of the island from the mainland, or how far from the mainland to the small island. That is the community structure. They have recognized the dispersal, but we don't have the mainland now. The habitat is being fragmented, so that we have a multiple habitat patches. They are interlinked. In these situations, we have another theory, that the metapopulation theory framework they have recognized the migration patterns, but they deal only one or two species. Suppose the migration depends upon the availability of unoccupied site in the other area. But this metapopulation framework basically deal about only the one or two species. And another thing is, species do not typically exist. Assuming that this is the process, metapopulation thing, that they have a unique niche for each species, but that is not the right, because I am influenced by the other species also. So therefore we are in the community ecology. We are looking. We are moving forward to improving the understanding of community ecology in larger system, that is the state of the local communities, by combining the two theories, that is the metapopulation and the community ecology. And in the framework, we have the models. We have, that is the past dynamic model, species sorting model, mass effect model, and the neutral model. So briefly, I'm going to explain this. What are these the model? The neutral model says that community are different among the patches only because of the dispersal limitations. If they have every species has equal fitness, so they can individually have equal ability to settle and succeed locally. But these community are different from this community, maybe because of the dispersal there is a limitations. This is the neutral model, where the past dynamic model is recognize the competitions. If they have, and also the trade-off between the competition and the dispersal, those who are highly competitive, they are not able to disperse very well. But those are inferior species, they have a large dispersal ability. So, but the habitat are all the environmental condition, all the among the patches are the similar. And similar in the species sorting model. Species sorting model is assumption that the heterogeneous environmental conditions, but the species sort by themselves and they persist in its favorable environmental conditions. So where is the mass effect model? Is the same thing as the species sorting model, but there is the influence of the high migration or the dispersal. So this is not the good quality. So this is the good quality habitat. So the growth rate is very high, and they are giving to all the species to this community until they but they cannot maintain their populations or species composition. So always giving the source and giving the mass species into another island. These are the general the models. So therefore, basically the community is structured two processes. One is the local environmental processes, that's the niche-based process, and the regional species process, that's the neutral process. Is hardly divided. We have this neutral process, the Stephen Hubble, that the theory is highly divided. Because why is it divided? Because we have a mixed results regarding which process, either regional or environmental, are important in structuring community. Because we have among the 158 published data set, 50%, more than 50% of the data set have the variation in the community composition is explained by both process, either by niche-based process or the neutral process. So what are the mechanism? Why mixed results? Underlying mechanism what? Still we don't know. So basically in the models assignment, we assign the based on the Metacommunity model based on the relative importance of space and the environment. If environment is very weak, so neutral process, the niche process is very weak, but the neutral process is very strong. That in that situation we can give the neutral or the patch dynamic model. Whereas the strong, both environment is very strong, and then the dispersal is very strong. That means all the species goes from one patch to the another patches. That is called the mass effect or the species sorting. And when the environment is very strong, the heterogeneous conditions, but the space, there is no effect of the space or the dispersal, that is called the species sorting. So, but that is the model we develop in the Metacommunity perspectives. But the extent to which this model applied to the real world is unknown. So basically why we are trying to feed the data on the entire regional set of species to one of the several Metacommunity models. We collected the data and tried to feed all the data in one Metacommunity models. And implicitly we assuming that all species operate similarly to over the same set of sites. But we have a difficulty to satisfactorily fit any single Metacommunity model to the natural system. Because as I mentioned you that all species operate, their assumption that all species operate similarly over the same set of the site. But species differ in their habitat use to the extent that such uniform treatment may be inappropriate. Because the species, we have suppose let's say the temperature is 0 to 25 degrees Celsius. Only the species they survive in only the 5 to 10 degrees Celsius. Might be the specialist, but those species in the same area, but another species may have there's favorable conditions that are suitable habitat. So there the distribution and the variation of habitat specialists most likely depend on habitat properties, whereas the habitat generalist, because everywhere is same for the generalist. So only if he get the ability, if he get the chance to disperse from one island to the another island, that means they can survive. So that means the dispersal is important for the generalist. So then the question comes, can Metacommunity system as I mentioned is a system composed of discrete habitats, exhibit an internal differentiation of structural process. Within a community, within the diversity, do you have the different processes? That's the central point of my research. And another thing is, we always assess the relative when assigning the model. Always we assess the relative importance of regional process and the local processes. Basically rely on snapshot data. But we know as a community ecologist, the community composition always changes over time. Therefore, the snapshot patterns may be insufficient for producing this consistent picture of meta-community dynamics. And another thing is the species richness. The greater condition when the one community and the another community, the one community has the very biodiversity is very low, and another community has the very biodiversity high. Those communities which are high biodiversity, we say that they're very stable, but population variation is very unstable. Therefore, that we do not expect the same pattern of the dynamics of any one species in two different habitats. So maybe there is a greater contingency in higher species richness in the meta-community framework. So that we are, therefore the differences in meta-community dynamics due to richness may introduce variation into the performance of applicable of meta-community. So basically I am looking in the three aspects, and they are my supervisor. While I was doing my PhD, they are my PhD supervisor. So one is the specialization, whether there is the fundamental error in meta-community theory or not. And another one is the lacking temporal patterns and the greater contingency in terms of the species richness. So let's talk the first thing. The question is, can meta-community system exhibit an internal differentiation of structural process? My prediction was habitat specialists governed by environmental factors or the niche processes, whereas the habitat general is governed by the spatial factors. This I did the research in Jamaica, that is the Caribbean country here, and this is the study area. In the Jamaica here, and then the Discovery Bay Marine Laboratory. And this is the aerial view of the test system rock pools. This is the very small, and all the blue are the rock. So where we sample. For the 10 years, we sample all the invertebrate species from this rock. So these invertebrate species from a rock pool, meta-community is a model system. So this one, the close view, this is the systems. So here is we use the different, the invertebrate species, and we have 49 pools within the 25 meter radiation, and each have a discrete, the boundaries. All the have we can consider as an island. And then we have a 10 years data, annual survey data, and the generation time for this species is 10 to 20 days. So within a year, almost 300 or 350 generation we can find the generation within 1 year. And then another one is we have 71 species, more than 300,000 individuals we have. And the volume is also the environmental, the gradients from. We have a homogeneous and heterogeneous. Environmental conditions are varied. And then the water are maintained mostly by atmospheric precipitation, and very occasionally wave expels water from the oceans. So we have a salinity to the fresh water, and also the environmental variables. So they are very heterogeneous, and each island, each rock pool considered as one island. So for the habitat specialization, I use. So we need to categorize we need to calculate which species are the specialists and which are the generalists. We use the Levin's methods. Basically, why we use the Levin's method? Because here is the problem in terms of, if we use the environmental variable to find the habitat specializations, that environmental variable, again we are going to measure, to find whether this is a regional effect or the environmental effect. There might be the circularity. Therefore, we did not touch that one. We look the uniformity of the distribution of species abundance among the habitat. There is no effect from the space and the environment. So only whether if they are uniform, all the 49 pools, that means they are generalist. If the sum of the habitat, they are like this and this one, means they are generalist, specialist. So we look the uniformity basically using the Levin's approach. And then once we calculated, then we have the habitat specialist, group of the habitat generalist, and combining of both group of species. And the environmental factors, we use the pH, temperature, different environmental factor, and the spatial factor. We did not have the dispersal, but we the distance is our proxy of measure of the dispersal. And all this, the environmental variable, we use the multivariate analysis to perform this, to answer these questions. What we found here, basically this is using all species. We can give it gives the signal here, the neutral models. So every year is the neutral model or the pH dynamic model. In the some year is species sorting and mass model, but in the majority is the neutral model, meaning that for this all the species, the dispersal is more important in the governing factor to structure the community in that the meta-community. In the same thing is the generalist, it gives more the strong signal for the using the dispersal. Here the more in the neutral model or pH dynamic model are more stronger. But looking at the specialist, we can see here the environment is all the time environment is important. So there is a neutral model importance. The neutral species sorting model is very strong for the specialist. What we can see here, using for this all species, this is environment, this is the space dispersal. Environment is low, the importance of the to structure the community. The environment is low for this when we look all the species. But when we go to the species, as I mentioned you earlier, there is shifting. So this goes up and this goes down. So the generalist in this species, it shows that there's two different processes going on in the communities. So the conclusion is meta-community can exhibit an internal differences of structural process with the habitat especially governed by environmental factors, but the habitat generally is governed by the spatial factors. Another is the static or the dynamic lacking the temporal patterns. So the perception of importance of environment and the spatial factors differ between the two approaches, because as I mentioned you that the community dynamics always changes over time. So the static approach and the approach that relies on the dynamics, inference will not be the same, will be different. That is our hypothesis. But the problem is, how do we measure? Because we have the multiple habitats, and also we need the temporal patterns. So how do we measure this one? We found that the spatial population synchrony, that is the index of population dynamic that tackle the aspect of space, as well as the temporal change of species abundance in multiple habitats. So what is the spatial population synchrony? So if we have two habitats, two patches, if the population fluctuation are correlated, that means the high synchronized. Their population fluctuation high synchronized. That is called the population. That can be, there is a different way. Population will be synchronized either by if the environmental conditions are correlated, then the population can be synchronized. If we have a dispersal, then the community will be synchronized. Or if we have a predator in both habitat, then the population can be synchronized. So these are the mechanisms. So our premises is here, environment constant habitat specialist by definition, more than generalist, and where the dispersal equalize the population size. If we have a two habitat, the two community, if they are the community are synchronized, that means due to the environment, then we can say that is the environmental factor important. If the community are synchronized by the dispersal, that means the regional process dominate on the population variation. So equalizing this is the two process, population A and population B. So we expect that is important for the generalist. So our hypothesis is generalists and the specialists should be synchronized by dispersal and environmental factors respectively. So what we found, here is the population synchrony, the y-axis is the population synchrony, and the environmental synchrony, y is the axis environmental synchrony. If those habitat, the population synchrony is higher in those habitat where environmental correlation is higher, that means slope will be very high, strong. R square, let's say R square is 1. But if there is no relationship, that means the slope will be very low. So, and then the slope, so we plotted the slope between the population synchrony and environmental synchrony, and this the niche breadth. The niche breadth is the smallest niche breadth is the specialist, and the bigger niche breadth, that is the generalist. So higher, the higher value is the very strong relationship, and the lower value is the there is no relationship, or is weak relationship. What we found here, so what we found, the effect of environmental synchrony are higher on habitat specialist than on the generalist. So what we found before for the using the static approach. Now comes to the dispersal. So if those communities which are highly synchronized, they can be synchronized, those which are very close by. So that means if it is go further, that means distant decay relationship, right? So if we go further, that means the relation will be weak. But in the closer, relation will be very strong. Like this. So what we found that all the species have, all the species have the negatives. That means except few species. That shows that the population are synchronized in the closed habitats. But the generalist and the specialist little differences, because the specialist here is a very high variation here. For this, these are the specialists, very high variation. Some species need, the population are synchronized. Those, for some of the species population are synchronized, those who are close by, and some species don't. Also sometimes the very negative or positive. So there is a high variation. So it indicating that the distance effect in general not important for them, for the generalist species. And the effect of species richness. So as I mentioned you earlier that if we have two communities, one is this community only have a monoculture and has a multiculture diversity. So population can decline under the unfavorable condition here, but may recover to the initial condition once it gets favorable condition here. But we cannot expect this here, because it's in a contingency. That is the compensatory hypothesis. Because if the population goes down of some species, that species can be recovered by the other species. So we will not expect the same the dynamics in the two communities. Therefore, the greater contingency can disturb the synchronization of population among the communities. So our prediction was the population synchrony in a community with fewer species would be higher, because as I mentioned you, the monoculture, they can, but there is no any biological effect. But in the higher species richness, there will be a biological effect. So we did very simple experiment. So we in the beaker, what we did here, that we introduced the different number, the environmental variability here, control, low, high, and here is the species richness, different species richness. The high environmental variability is, we did this week 0 ppt salinity concentration, and the next week 6 so we change every 7 days. So but here is low environmental synchrony, low environmental variability has the 0 ppt and the 3 ppt. So we give the 3 level of treatment. And another one is, this is the 4 beakers here. And how do we create the meta-community? So we took the sample every week. We took the sample 10 ml from here, 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 and we mix them and put back the 10 ml for each community. So we created the meta-community. And this is the 2 species. and this is the 3 species and the 4 species. So now we have ability, to assess the importance of environmental variability and the species richness. So what we found here, regardless, here, this is the low control, low and high environmental variability. Very high. Population synchrony is very high if those communities which are exposed high environmental variability. But look, here is the generalist that does not affect. But regardless of the species richness, there is the effect. We can see that the species richness. This is only the monoculture. This is the 2 species, 3 species, and 4 species. We can see that there is a main effect of the species richness and the environmental effect. So the population synchrony is higher for the intermediate species and the specialist, irrespective of the environmental variability. And there is no interaction effect, meaning that there is the main effect of temperature and main effect of environmental variability and the species richness. So what are the conclusion here? So the degree of synchrony. So if we have the climate change, we do not expect the same level of we have 2 different communities, meta-communities of the landscape. We do not expect the same level of population, the dynamics. So that they perceives, it depends upon how a particular species perceives the strength of environmental fluctuation. One paper that is published in the Nature in 1998, they did look whether the environmental has the effect. There is no any dispersal. The 2 islands. They look what the effect of the environmental condition, environmental correlation for this species, and they found that only the population synchrony was only the 0.52. That was able by only when the population synchrony was 9. So our expectation, their expectation was, if population are synchronized completely, then this population will be synchronized, but does not. Meaning that how this may be, if they have looked the other species, might be they would have the higher species richness, a higher population synchrony or the lower population synchrony. It depends on how a particular species perceives the strength of environmental fluctuation. So I am not going to talk to this one. Regarding within the periphery of this research, we did a couple of other publications. So in the very in a short form, what we found in all this research, the meta-community system can exhibit an internal differentiation of structure process. And then Hubble theory, the stuff I mentioned is why the mixed models, why the more than 58 published data set, they have a mixed model. Most likely, given our results, it is possible that the system studied by other authors had a different ratio of specialists and generalists. That's the answer. Now goes to the second the topics about whether we can improve the biodiversity model in regarding the climate change. We know that we have the issue of global warming. So the biodiversity is expected to be decline, mainly as a function of the decreasing regional precipitation and increasing evapotranspiration. So this is the mammals richness of the global we have the 258 species. We can see that how do we detect this species richness? The species richness in the global and the regional mammals are explained by actual evapotranspiration. The energy and water, always we thought this, this one, energy and water are the driver in explaining the regional and the global pattern of species richness. The water and the energy are the most important parameter to explain the species richness for the mammal global species richness. But we don't have the actual we don't have the actual evapotranspirations for the future. So lacking the actual evapotranspiration, using the several parameters to predict the biodiversity in the future, And we know that, the standard approach is we use the mean temperature and mean precipitations for the distribution and species richness. And we use the sophisticated curve fitting procedure, but that doesn't have any reality. So, and we give the other, we propose the other alternative hypothesis, and also the habitat heterogeneity, surface area, regional evolutionary history. So this other alternative hypothesis we have actually because of, we could not find the clear patterns using the mean temperature and precipitation and other environmental variables. So this work, I work with Stephen Campbell from the University of Quebec at Montreal, and Pedro Perez Neto, he is also at University of Quebec at Montreal, and Jonathan Davis from McGill. So I worked this project with them, and looking at how do we improve the biodiversity models. We know that the climatic variables are not uniform in homogeneous as well as the temporal patterns. So the temporal pattern in climatic variables vary in space, and it is considered to be an important driver in species composition and the stability patterns. But we are not looking at the temporal patterns of the temperature. Instead, we are using the mean temperature and mean precipitation. That is the, that's the problem. So, and our research question is that does time series component of climatic variables, we decompose the climate, temperature, and precipitation into different components. The linear component, seasonal component, cyclic, and the residual. Whether if we use this variable, can we improve the model, and then improve the performance and bring new and future insight on species richness modeling? And if yes, then which area would be more sensitive in terms of the species extinction, immigration, and the turnover? And then these all are the taxonomic biodiversity, but whether the everyday knowingly or unknowingly we are losing species, right? But we will notice this only if the ecosystem is not functioning, it's collapse, right? So that means we have to look the functional diversity or phylogenetic diversity. The phylogenetic diversity, if there is a more diverse, they give the ecosystem is more functional. So does the climate change affect on functional and phylogenetic diversity? That are the main the research questions. So for this work, we use the species, that is the distribution pattern of 4,300 mammal species, and it is 1 degree grid cells. And then we use the time series climatic variables from the 1961 to the 1999 for the present, and then for the future to 2061 to the 2099, that is 0.5 degree spatial resolution. And we use the time and precipitation, temperature and precipitation 3 scenario and 8 models, the 24, so let's say altogether 24. And the other variables for to monitor to develop the model, soil texture data and sunshine percentage and digital elevation model and actual evapotranspiration. And then another variable we use that the major scale microclimate, because 100, the 1 degree means almost 100 kilometers. So in order to detect to catch the microclimate, we use the interaction of the mean temperature and the range of elevation in a grid, because we have a digital elevation 1 degree. So what the range of the elevation from for the 100 grids? So we calculate the range and then mean temperature. And the variables, the temperature, we decompose into the different parameters, cyclic parameter or the linear, whether we have a linear patterns of the temperature or we have a cyclic, or the seasonality, or the if there is no any if the temperature is not explained by anyone, then the residual. So we use the 40 years data per monthly mean. So that means 40 times 12, 480. So we use the data to decompose the temperature and precipitation. And all together, that is only the one land only, and then we have 32,000 grid cells. It took me almost 15 days to do the analysis. So I did in the MATLAB. This is the MATLAB. I did in MATLAB, and then it took me 15 days for the global scale. So what we found in the temperature, this is the real, this is the current temperature, And what we found that this is the linear. We can see this is in the equator area. The temperature is explained more by the linearity, where the seasonal in the northern and southern part is more pronounced, seasonality more pronounced, is obvious. And the cyclic, we can see that the cyclic patterns in the equator region and also the residual, maybe because of the seasonality, he is not here in the seasonality explained very well. This is 0 to 1, means the percentage of variation explained of temperature. 0 means nothing, 1 means 100%. So almost the seasonality in the north and south is temperature explained by the seasonality. Same thing in the precipitations. In the precipitation, we did not find the seasonality here. This is very common. We know the precipitation does not have a uniformity. So there is no seasonality, and the linearity is very small. Almost there is no cyclic. Little bit we can see some cyclic patterns in the precipitations. So what we found here, this is the species richness. Y-axis is the species richness, and looking the relation between the species richness and this variation, the temperature and precipitation. This is the temperature, linear, cyclic. This is the 4 component, and for the precipitation we did not see any significant relationship. We can see that because of so many the sample size, we can see, but it's very few variation explained by the precipitations. So what we found here, so our objective is to know whether the mean temperature and precipitation explain more, or these are the parameter explain more. So we did the modeling, and then we looked we did the variation partitioning. Here is the 3 aspect, 3 scenario. Using the actual evapotranspiration, what we have a data, but in the future we don't have a data. Using the inmounted the AET, and this is the temporal components, what we derive now. And this traditionally we are using mean temperature and precipitations. So if we use only this model, we will explain only the 69% of the variation of the species richness in the global and the regional scale. Whereas if we use the AET, there's the scenario, in the model, we will use 71. Whereas the temporal component explained significantly very high, 80% significant. How they, whether they are, whether the variation are shared among them, we can see that, here is the actual evapotranspiration and the mean temperature evaluation, this one and this one. Unique contribution of actual evapotranspiration is 14%, where the temperature, this one is almost the 50% are shared between the 2 variables, and only the temperature are explaining the 2%. So residual value are 23%. We are not able to explain 23%. Whereas the TC, the temporal components, what we derive using this one and this one, we are able to do the 82%. Where is the 77? That means, but in the future we will not have the AET. Only we are struggling to make the different models, trying to fitting the data. Only we for this one. But if we include this one, already we have we can do this the temporal components, and then mean and temperature we can explain almost the 82%. Only we are losing 3%. That if we use everything, AET, TC, and the MTU, we will have 85%. But in the future, we will not have that. So that if we use, we have 82% explained variations. So we based on this the model. So we thought, using our what I showed you before, using this one, the model, we predict the species richness for future, and also we develop the species distribution model for each of 4,030 species. And what we found, this is for only the North America. This is the observed the species richness, and this is the ratio, future, the present divided by the future. So we could do the subtract, because they does not give, because some of the area have only the one species and some of the area have the 250. Does not give the sense. So we did the ratio, and 1 means perfectly the same species richness. And if this is less than 1 means decrease, species is decrease, but is greater than 1 means species increase. We can see that here the green is species number will be the same, but this the gray little bit species will be decreased, and other the yellow is species will be increased. Let's say this in close view. So almost only the 10% of the area will have the species same number of species richness. About 30% will have the less number of species. And the another one is, where do we, that is using only the species richness. Now looking for each species, where do we gain the species or we will lose the species? As we know that the polar regions, this is the polar region, this map is based on the main climate Köppen-Geiger's classification, right? So we can see that the polar, almost 27% we can gain the species here. And same thing in the snow area, will also the 22% we can gain the species. And very low species we will gain here, with the equator. Will not. Here is the assumption that there is no restriction of the dispersal from one pixel to the another pixel. And the species loss, equator, we will lose almost the 15 in the 16%, but we are gaining because most likely species going up, right? So that we will lose the species 50%. Turnover here is a turnover or very high turnover in the polar area, and in the snow area is a high turnover, but in this the equator area very low turnover. So, and another question was, does the climate change affect on functional and phylogenetic diversity? So we have this phylogenetic diversity of the the communities. So if we have we have a climate change, this is the filtering, is like a filtering. If community change, if the is climate change affecting on the species, so how in the future whether the species within the community are more closely related. If species are closely, the phylogenetically, if they are closely related, means that something about the environment is important, so that the climate change is affecting that species. If we have, the more distinctly related, different, the phylogenetic diversity, different the traits. If the different traits, the species with a different phylogeneity, they coexist, meaning that environment is not important. There might be the the competitions or the biological factor the importance, right? So if species with the similar traits coexist more commonly than expected by chance within a community, then the climate change or the habitat attributes driving force. But in the second step, that the resources are limited, or biological effect prominent. So this is the current, what we found here. Still I am working for the future, I have not done it. But the closely related here, in Australia and the northern part, is environment is very important, very harsh. In Australia, it's very harsh conditions. So there are more closely related species there. But the other places, here is the more distinctly related species will be there, are there here. So it shows that the environment, if once we done for the future, we can see whether the climate is really affecting the phylogenetic diversity or not. And the summary, what we found that the temporal variation of the climate variable are good predictors in explaining the large scale variation of species richness. And on an average, 12% of species lost per grid. And several areas have been projected to gain the new species as a result of range expansion, assuming no dispersal limitations. And due to the combination of both potential range contribution, contraction and the range expansion on an average turnover age about 50% we can expect in the 2080s. So another one, I'm not going to discuss this one. What we did for the last 2 years, is very big group. So I work with the UQAM, McGill, and the consortium on the regional climatology and adaptation to climate change. We developed the methodology to monitor the biodiversity in function of climate change for the Quebec province. That was work for the Quebec government. So maybe in the next time I will give a talk this one. And another one is recently is in press about I developed the habitat models. This is the Lake Erie. This is the Ontario, and this is the USA. So we have, the walleye is a commercial fish species, and we have income from the walleye about $1 billion annually. So we have a quota systems, but that quota system is not so standard. There is no scientific judgment. So I developed the model, and they are using this my model for the quota allocation. And another one is this is more methodological. Suppose in community ecology, in biodiversity, species area relationship. If area increases, species will be increases, right? Same thing for here in the distribution model. Do we, what will happen? What the performance of the model if we increase the plot size, collecting the sample in the field? So I did in the Namibia, in Africa. So I have to acknowledge, I have to acknowledge so many, but those who helped me to accomplish these projects. So basically, Yurek Kolsa, he's my professor, and Carl Cotten and Matthew Leavitt, he's the renowned scientist in community ecology. And David, he's recently he got the job in Yale University, and Pedro Perez, he was my previous postdoc. And Jonathan Davis is also the McGill University. Stephen Cavell is also the professor, and Lisa Manny, he is also the professor at the New York. And then the funding resources, I got the funding from Ontario OGS, Ontario Graduate Scholarship, and the Quebec McMaster University and NSEC. Thank you.